we're going to be talking about or learning about a set of issues around chromosomes, condensates, and transcriptional control. This is occasioned, of course, by the enormous changes that happened in our view of these processes inside cells over the last 5 to 10 years. And it is a pleasure to start off with Stephanie Weber from McGill University, who's made her wonderful contributions to this class of problems. She started out thinking about intracellular organization in bacteria, and moved to thinking about the problem of condensates as these issues were just emerging, and understanding how the sort of droplets that constituted the things we used to think of as organelles that turned out not to be membrane bounded, how those scaled in size with the size of cells, and has now sort of merged these interests and is thinking about condensates in bacteria, and in particular in the context of transcription. I'll keep an eye on the chat. If something comes up that really seems like we need a pause, I'll interrupt. And if not, I think Stephanie has sort of programmed in a few natural pauses where we can take questions. And so with that, Stephanie.
Okay. All right. Thank you, Bill, so much for the invitation and opportunity to pseudo see you all. I'm going to talk today about condensates in bacteria. So this is a new story that just came out from my lab, and then I'll try to connect it to sort of larger questions of transcription and that the other speakers will pick up on. So typically when we think about cells, we think of eukaryotic cells, and this is an electron micrograph of a mouse cell, and what you can see are lots of membrane-bound organelles, right? So there's compartments that are surrounded by membranes. There are all sorts of different sizes and shapes, but all of them have the common feature of having a membrane around them that creates a diffusion barrier that spatially separates things inside the organelle and things in the cytoplasm. And so this allows every organelle to have a distinct biochemical composition from the surrounding cytoplasm, and that facilitates whatever the specialized function is, whether it's maintaining the genome, packaging, sorting proteins, making ATP, whatever. And so until about 10 years ago, the assumption was that inside the cytoplasm, where you don't have membrane compartments, that molecules are free to diffuse around and mix. And depending on what the function of these particular molecules, that could have adverse reactions. And so for probably 200 years, membrane-bound organelles were considered the unit of intracellular organization. And because bacteria don't have them, it was assumed that bacteria don't have organelles, period. And so I would like to remind you guys that eukaryotes, which we are all made up of, actually represent just a really small twig on the tree of life. So the vast majority of life on Earth is prokaryotic. And in contrast to this cell that has lots of compartments, prokaryotes are relatively unorganized. They don't have membrane-bound organelles. You can see a little bit of separation between the electron-dense ribosomes in the cytoplasm and the electron sparse nucleoid here in sort of white. But there are no discrete organelles. And so the question that I had was how do bacteria sort of organize and self-regulate all of these biochemical processes that eukaryotes use membranes to do? And so despite their lack of membranes, many molecules inside prokaryotes still adopt a non-uniform localization pattern. So proteins, so this is the FTSZ, which forms the Z ring, which creates the septum during cell division, localizes to the mid cell. Different chromosomal loci occupy specific locations in the cell. RNA and lipids also occupy specific locations. So the cell is not a bag of enzymes where everything is uniformly distributed, but the mechanisms that sort of allow cells to put specific molecules in specific places are still unclear. And so in bacteria, there's a couple mechanisms that are known. So I just said that bacteria typically don't have membranes, and the first example I'm going to give you is membranes. So magnetosomes are invaginations of the inner membrane, and they form sort of membrane-bound organelles that line up and allow certain bacteria to align in the magnetic field. And this allows them to orient in sort of the water column, for example. There are other membraneless organelles, such as storage granules, and these are sort of aggregates of plastic, essentially. Long polymers that are insoluble, and they form sort of spherical granules. And then the best studied case are what are called bacterial microcompartments. And so rather than a membrane, these have a protein shell made up of sort of icosahedral proteins that form in these large, sort of very geometric structures. They almost look like viruses. And these create a semi-permeable barrier. So the best example is carboxysomes, and that allows rubisco to concentrate inside and carry out carbon fixation. And so those are just some of the organelles that do exist in bacteria that have been well characterized. But there are still a lot of sort of open questions about bacterial cell organization. And so recently, as Bill alluded to, phase separation, liquid-liquid phase separation, has sort of exploded on the eukaryotic cell biology scene. And this is a process where proteins and often RNA condense to form discrete clusters or local concentrations of protein that have a different biochemical composition from the surrounding cytoplasm, and yet they don't have a physical barrier separating them. And so this is an example of germ granules in the C. elegans embryo that Cliff Brangwyn, my postdoc supervisor, took when he was a postdoc in Tony Hyman's lab. And what you can see is that in the anterior of this embryo, these clusters of protein sort of disappear, and they appear and grow in the posterior end of this embryo. And so this is thought to allow the cell to sort of partition cytoplasmic components asymmetrically during division to determine cell fate in a developing embryo. And then even in mammalian cells, so in our cells, when we are exposed to stress conditions, so for example here, the experimenters added arsenite to the cells to stress them out, and that induces very rapidly stress granules. And you can actually see examples of some of these fusing. And what I want to emphasize is that this is a reversible process. So when the stress is removed, these organelles, because they don't have a membrane, are very dynamic, and they can assemble and disassemble in response to either developmental or environmental cues, and may allow the cell to respond much more rapidly than changing sort of membrane synthesis would be. And so just as a quick primer on liquid-liquid phase separation, the idea is that you have two states. One is where the molecules that make up these organelles are mixed and dissolved in the cytoplasm, or in this case, the nucleoplasm. And then there is another state where they are phase separated. And so the green molecules here form the nucleolus, which is what I studied as my postdoc. And so here they're dispersed in the nucleoplasm. Here they're condensed, and they often, and as you saw in the previous slide, form spherical droplets. And so these droplets are reminiscent of lava lamps, if you want, and they behave like liquids. And so because they fuse and they round up, etc. And so we can think about this as like a liquid mixture, right? So the cytoplasm or the nucleoplasm is just a complex mixture of molecules, and they can mix like oil and water in your beer. I guess it's a little bit early for that, but whatever, if you want. it's 2020. Oil and vinegar can separate into two distinct phases, even though there's not a physical barrier, diffusion barrier separating these two phases. And so this picture is emerging of cells as sort of formed of a variety of different liquid phases that each have its own not only chemical composition, but also material properties. So some of them are, very fluid and rearrange and run very quickly. Some are more viscous or even viscoelastic. Sort of an open question in the field is how the sort of liquid-like properties for these membraneless organelles allow them to form and carry out their particular function. So I want to go back to this question of bacterial cell organization. And actually, before I go on, Bill, if there are any questions at this point, I'm happy to pause.
No, I think we're okay.
Okay.
We can encourage people, but
yeah, if you have questions, please put them in the chat or ping Bill. Otherwise, I'll go on. So the question that I had was, since there are sort of numerous and ever-growing numbers of examples of phase separation in eukaryotic cells, do bacteria also use this mechanism for cellular organization? And my rationale was that they might actually be even more common or more sort of functionally essential or functionally important in bacteria because they don't have membranes to carry out this function. And so maybe this is the sort of only or best alternative mechanism. What I fail to remember or appreciate is that bacteria are freaking small. And so actually, perhaps one of the reasons that they haven't been described in bacteria yet is that it's really hard to study at these small length scales. So anyway, this is what the sort of first project that I tried to pursue in my brand new lab, which may not have been the brightest idea. But anyway, okay. So taking a step back, there's actually some evidence of sort of phase separation in a different sense that had been described 20 years ago, actually the year I was born. And so if you look at bacteria, so here you can even start to see it. So the ribosomes, which are these black circles, are excluded from the nucleoid, which is this electron sparse sort of white region here. And that can be described as a separation of phases, right? So here they looked at the refractive index of cells, and there is a different refractive index inside the nucleoid than there is in the cytoplasm. And so that, basically tells you that the cytoplasm and the nucleoid have different physical properties and different sort of phase behavior. So that can already start to organize, sort of separate the cytoplasm from the nucleoid. And then the specific example that I was really interested in is looking at RNA polymerase. And so this comes to Bill's point about transcription. But the reason that I was interested in this problem is because bacterial RNA polymerase is roughly uniformly distributed throughout the nucleoid in slow-growing cells, but it condenses into bright clusters in fast-growing cells. And other evidence had suggested that these clusters either co-localized with ribosomal RNA genes and or were at least correlated with upregulation of ribosomal RNA. And so in my mind, these look a lot like a bacterial nucleolus, where Pol-1 in eukaryotic cells clusters around rDNA and synthesizes, ribosomal RNA to form ribosomes in eukaryotes, and maybe the same thing is happening at a much smaller scale in bacteria. So this was the initial observation about almost 20 years ago now, but the mechanism by which this sort of redistribution of the polymerase inside the nucleoid was unclear. And so, so my postdoc, Anne-Marie, and undergraduate Graydon started looking at this problem, and they did a very simple experiment, partly because it was a new lab and we didn't have the equipment to do this more sophisticated, but they did an outgrowth experiment. So they took an overnight culture of bacteria, they diluted it, and then they just took samples of cells at different time points from the outgrowth. And unfortunately, we couldn't do this at the time in live cells because the clusters that we see in fast-growing cells are so sensitive to the environmental conditions that when Anne-Marie took a sample out of the incubator, ran to the microscope and mounted them on the microscope, they were already gone. And so she sort of had to resort to fixation. And then what we did was we looked at the distribution of the fluorescence in the cell. And so in stationary phase cells, you get a roughly uniform distribution of we're tagging RNA polymerase, a large subunit of RNA polymerase. And then when they enter log phase, you start to see these discrete clusters, and the background, so the number of pixels in the background that are dark gets larger. And so, well, the way we quantified this was by looking at the normalized fluorescence intensity histogram of all the pixels in a cell. And so, so down here are the really dim pixels, up here are the really bright pixels. And what I want you to notice is that moving from stationary phase to log phase, there's a shift in the distribution of pixels from bright cells to dim cells. So, bright pixels to dim pixels, sorry. And so to compare this with a control, we looked at HUP-A, which is a nucleoid-associated protein. So it binds roughly non-sequence specifically everywhere in the nucleoid. And here we get a very uniform distribution of pixels. And then we also compared it with LAC-I. So LAC-I binds to an array of the LAC operator. And so it binds to a specific single site, or multiple sites because it's replicated, in the nucleoid. And so here we see a very clustered distribution where there is a lot of pixels that are really dark and just a few really bright pixels. And so comparing these distributions, we set an arbitrary threshold of 50%, and we said, okay, the degree of clustering is the number of pixels below this threshold. And so for a dispersed distribution, this is a relatively small number, and for a cluster distribution, this is a relatively large number. And then we could look at the change in the distribution over time. And so this is an example of the time course. So 0 is they've taken cells just out of stationary phase, and then over time the cells grow, and the polymerase forms these clusters, and these clusters peak in mid-log phase at around 120, 180 minutes. And then as the cells re-enter the next stationary phase, the cell size gets smaller and the polymerase redistributes and disperses throughout the whole cell. So first of all, this is a reversible process. And so here we can see that in stationary phase cells, there's a low amount of clustering. It is roughly comparable to what the HUP-A sort of negative control uniform distribution. And as the cells enter log phase, it increases, and then it slowly decays again. And I just want to point out at the 60-minute time point, there's actually a bimodal distribution. So some of the cells have formed clusters and some of them haven't. And so it looks like there is a relatively fast or maybe discrete transition from a dispersed distribution to a clustered distribution. So this is an LB, which is a rich media where the cells grow very quickly. So in log phase, these cells double about every 20, 25 minutes. And if we look instead in a minimal media where there's limited resources, these cells grow more slowly. The size of the cell doesn't become as large, and the clustering does occur a little bit, but it's nowhere near as discrete as it is in the rich media. So here again, we can look at the clustering over time, and there are two things to notice. So first, the maximum clustering is lower than it was in the rich media, and second, it's all shifted because the cells grow slower, and so there's a delay. Okay, so we can quantify clustering, and so we, as I mentioned, comparing LB to M9, the percent of clustering depends on the doubling time or the growth rate of the cells. So in LB at 37, there's a high degree of clustering and a really slow, or sorry, a short doubling time, fast growth. And in M9, there is less clustering and a long doubling time or slower growth. And at intermediate growth rates, so if we grow bacteria in LB at 30 degrees Celsius, then we get an intermediate both degree of clustering and doubling time. And I am going to wildly speculate here. So I know this is 3 points, but I was really intrigued. Oh, sorry. Before I speculate, these results, sort of a lot of clustering at fast doubling times and fewer clustering slow doubling times is consistent with single molecule imaging. So, so previous studies had shown that that RNA, by looking at counting up the location of individual RNA polymerase molecules in the cell, form clusters, and there are more and larger clusters in rich media and fewer smaller clusters in minimal media. Okay, so back to my speculation. These 3 points roughly make a line. And the reason that I was really intrigued by this is because there are what are called bacterial growth laws, which are these empirical relationships between the ribosome content, or the RNA to protein ratio in cells, and the growth rate. And so as you increase the, as you change either the growth medium, the growth temperature, antibiotic concentration, anything that changes either nutrient quality or translational capacity that affects growth rate also results in a linear change in the RNA to protein ratio. And so this, to me, looked like maybe these clusters facilitate the synthesis of ribosomes and maybe could explain this linear relationship of the bacterial growth law. But that is wild speculation based on 3 data points. Okay, so before I move on, there's one question in the chat, and I'll pause here if anyone else wants to ask a question. Bill, do you want me to read from that, or do you want to moderate? I don't know how this goes.
I don't think it matters very much. So one question is about estimating the size of the clusters.
Yeah, so we have not at all. So other people. Achilles Kapanidis in the UK has done single molecule imaging. And so they count up, or they find the location of every single polymerase in a cell. So that's this top image, and each red spot is a single molecule. And then they do a clustering algorithm of sort of like a nearest neighbor, you know, how many would cluster together. And so that is their, this bottom image here. And so they just literally count up the number of molecules that they detect in a particular region of space. And that is their, so sorry, all of these numbers refer to the number of polymerase molecules assigned to that particular cluster. Our clustering is is not actually measuring the discrete size of particular clusters. It's just measuring the relative distribution of the polymerase inside the cell on a per cell basis.
Good. There's another question about issues of different gene copy numbers.
Yeah, so Yanni, that's a fantastic question, and I'll get to that a little bit. So yes, absolutely. The other thing that is changing between M9 and LB is the DNA is being endoreduplicated, and so there are more copies of the chromosome, and specifically there are more copies of over-enriched more copies of ribosomal RNA genes, because those are located primarily near the origin of replication. And so yes, the copy number of ribosomal RNA genes is much higher here than it is here. And certainly that could, I don't think it's the only thing, but that could certainly contribute to this difference. And I'll show you a little bit more data on that later. Yeah, it's a great question.
Actually, do those copies tend to be physically close to each other, or?
That is also a really good question. So it's a little bit of a controversial thing. So there is a paper that shows that, and I'll show it maybe in the next slide. There have been some reports that ribosomal RNA genes cluster, co-localize inside the cell, and there are some reports that they don't. And we have done preliminarily and not seen that, but yeah. So I think that's still an open question.
Good. Let me get the maybe simpler ones first. So there's a general question, I guess, about whether clusters are only proteinaceous or whether they also include RNAs. Maybe you could say a few words about that.
So they almost certainly contain RNA. So if we treat with rifampicin, which inhibits RNA polymerase, then they disperse. And in vitro, when you add RNA, the droplets of a transcription factor, not the polymerase itself, get larger when you add RNA and smaller when you remove RNA. And in other sort of condensates in eukaryotic cells, they've shown that there's an RNA dependence. So these, we haven't characterized that too super well, actually at all, in vivo yet, but I would be shocked if RNA wasn't present and didn't play a role in stabilizing them.
Good. There's a general question about how the phase separation is controlled, but maybe that's going to evolve. That question will sharpen at least.
Yeah,
I think
we'll definitely come to that later. I mean, the short answer is I really don't know yet. We have a couple of molecular candidates for sort of disordered proteins, but how it's regulated in response to growth rate is sort of an open question that my lab is looking into now.
Which is, I guess, related to the question of how phase separation is broken, as it says in the question, when molecules need to interact in other places. But I guess for the RNA polymerase in particular, it's not clear. Do they need to go somewhere else, or are they clustering in the place where they need to be? Yeah,
right. So Jie Zhao at Johns Hopkins has shown so they've done FISH for the rDNA and looked at these clusters, and they see some co-localization, but they also see some clusters that clusters still form even when you don't have rDNA present or even transcription present. So I think there's a correlation, but I don't think it's necessary.
Good. And then there's a question about, actually questions are starting to come more rapidly.
Yeah, I'm having trouble keeping up, so I'll let you moderate.
That's okay. Let me see if I can sort them for you. So is this specific to RNA polymerase, or do other DNA binding proteins do similar things?
That is a good question. So I don't think it's specific. I'm sure there are other DNA binding proteins that may do this, but it is not, and which I'll show you later, it is not a consequence of DNA binding. So there is something in addition to DNA binding that is driving these clusters to form. But it wouldn't at all surprise me if there were other DNA binding proteins that also form clusters beyond just their association with DNA.
And then there's a kind of general question about the viscosity and density of the cytoplasm, the cytosol. What I guess maybe to make the question a little sharper, you see the condensation of some droplets of protein, but do you know whether there's a kind of general is this related to any broader variations in the physical properties of the surrounding medium?
That is a fantastic question and something that we're interested in. So we've super quick and dirty N of 1 tried to do some EM or cryo-EM on these cells in the stage when they have these big clusters, and we couldn't see anything from that. So if they're there, I don't think the I think in bacteria the cytoplasm is so concentrated and dense already that I'm not sure that this is significantly, at least from a gross perspective, higher. I think it's more a compositional difference rather than a sort of molecular density difference.
Good. Okay, let's do one more and then we should let you get back on track. About whether RNA species contribute to the condensates. And so is it the same types of RNAs that are clustering together, and are they segregating from the others?
So here, I imagine that most of the RNA present is ribosomal RNA, so new transcription at these particular sites. But in other condensates and eukaryotes, the specific RNA can determine multiple things. One, it can be separated so that some condensates have certain classes of RNAs that are excluded from other condensates, and two, the identity and really the structure, so the secondary structure of the RNA itself can determine or can contribute to the sort of selectivity of proteins that go to the condensate and to the material properties of sort of like the viscosity or viscoelasticity of different condensates. So we haven't really started exploring that question in bacteria yet, but Amy Gladfelter has some beautiful work on that in what is it? Yeast, fungus, Ashbia.
Good. Okay, I think I'm going to myself and let you.
Okay. Yeah, great questions, and this is fun. Okay, so one of the questions asked a little bit about what is driving the phase separation, or sorry, what is driving the clustering, and the question also came up about DNA binding. So this is right on, right? Like I was super excited about this clustering, and I was, obsessed with phase separation because I just left my postdoc from Cliff's lab, and I went around telling people that, oh, this happens in bacteria, and everyone told me I was crazy because, oh, it's not phase separation, it's just that the polymerase is binding the DNA, like that's it. And because ribosomal DNA is the most highly transcribed genes in fast-growing cells, all you're seeing is a clustering on the DNA. And so one perfectly reasonable null hypothesis to explain this clustering is just that the polymerase is binding to DNA, period. I had the, favorite pet hypothesis that maybe phase separation is contributing to this. So LLPS is liquid-liquid phase separation. And the idea here is that some polymerases are for sure bound to the DNA and actively transcribing. But maybe there are either the polymerase itself or sort of other transcription factors that are able to form multivalent interactions and hold other polymerases, tethering them to this location, even though they're not directly bound to the DNA. So this was my hypothesis, or my two models that I was trying to distinguish. And so, to address the first one, and this kind of builds on Yanne's question. So there's these beautiful EM Christmas trees of ribosomal RNA genes, and the idea here is they break open the cell and they spread the chromosome out, and what you can see here so the backbone of this image is the DNA. Each of these black spots along the line here is a polymerase, and then the RNA are these squiggly lines coming out. And so, what you can see is that the polymerases are like bumper cars along this ribosomal RNA gene. They're really, really packed, to the point where if you count them up, there is one polymerase every 85 base pairs. So you would certainly see an accumulation of polymerases at these genes just purely based on DNA binding. Okay, so to make this a little bit more quantitative, though it's still kind of hand waving, if we look at the genomic organization of ribosomal RNA genes, like Yanne alluded to, there are 7 of them in E. coli, and most of them are clustered around the origin of replication. And so the E. coli genome is a circle, and it starts replicating at the origin, and it goes bidirectionally and stops at the terminus. And so genes near the origin of replication will be replicated first. And so once replication starts, there will be 8 of these, and so the number of RRN operons will increase and become more abundant in cells growing at fast growth. So each of these arrows is an operon that encodes the 3 ribosomal RNA genes, so 16S, 23S, and 5S as a polycistronic unit. And to follow up on Bill's question, there are so these 4 operons are almost certainly like co-localized in the cell, just because they're literally tethered on the chromosome together. So they're going to be close in space because they're close in genomic space. But the question was, are these operons also co-localizing with distal RNA operons that are not close in sequence space? And so Rick Gorse's group tried to look at the RRND and the RNB. So looking at this operon and this operon, and seeing, do they come together in cells? And he found that they did. So in slow growing cells and in fast growing cells, in all cases, he found a co-localization of a ori-distal gene, RN gene, and ori-proximal genes. So there is at least some evidence that RN operons co-localize in cells. Again, suggesting that maybe just binding the DNA is sufficient. And so if we do a real quick back of the envelope calculation, and this is an underestimate, but just as a minimal estimate, there are 7 operons And this is not counting the fact that they would be replicated. So this is an underestimate. Each operon is 5.5 kilobases long. And if we assume that there's one polymerase every 85 base pairs, then that would give you around 453 RNA polymerases bound to the DNA, to RNA operons that are clustered together in the cell. And so if we go back to the Kapanides estimates for the number of molecules that are in these clusters in rich media, then it's comparable. So they count, several hundred molecules in each of these clusters, and we are, as this super underestimate is roughly comparable. And so I sort of was a little bit disappointed in seeing this, because it looks like DNA binding is sufficient for clustering, and so maybe phase separation is not necessary. But more importantly, and I guess more fundamentally for the field of phase separation, and this goes for eukaryotes as well as prokaryotes, it's been really difficult to measure or to detect definitively liquid-liquid phase separation inside cells. It's really easy, or not easy, but it's relatively more straightforward to purify disordered proteins, to see droplets in vitro and to say, oh, that's what happens in cells. But actually seeing this and measuring this in vivo is challenging. And so what are the criteria? What could we look for inside cells? So there are typically 3 different experiments or criteria that people use to define a phase separated organelle. So one is that these clusters or droplets fuse, right? If they're liquids, if 2 droplets come together, then they should fuse and round up to form a large spherical droplet. They should have a spherical shape, and this is because the phase wants to, or thermodynamically, you want to reduce the interfacial tension between the surrounding cytoplasm and the droplet itself, and so that will drive rounding. And then inside both inside the droplet and between the droplet, molecules should be mobile. So you should be able to see molecular rearrangements inside and molecular exchange between a condensate and the surrounding cytoplasm. And so to do this in eukaryotic cells, people take time-lapse movies. So the movie I showed you of stress granules earlier in this talk, you see droplets that fuse together. You can look at the aspect ratio from images. So these are very spherical. This one is a little bit elongated because it's in the middle of fusing. And then looking at molecular mobility, the most common experiment to do is FRAP. So fluorescence recovery after photobleaching. And specifically, there are sort of 2 flavors of FRAP. You can do a full FRAP experiment where you bleach the entire condensate and then look at the recovery, and that recovery is from exchange. So external molecules that are external to the condensate coming in, or you can do what's called a half bleach. So here is a pea granule, and Cliff bleached half of this granule and then looked at the recovery from within the granule itself. And so you can look at both, with FRAP, you can look at both exchange with the external medium and internal rearrangement. So these are very common assays done in eukaryotic cells, but in bacteria we have a problem of scale. So in this C. elegans embryo, the embryo is 50 microns across. These pea granules are on the multi micrometer scale, and so it's possible to do FRAP on part of it. It's possible to see the shape of these things, right? It's well above the diffraction limit, and so you can look directly at fusion, shape, and mobility just by imaging with light microscopy. But what I want to point out is that the cell that I'm looking at is smaller than this granule. So trying to do some of these experiments on clusters inside sort of a 1 micron cell, and these clusters are absolutely sub-diffraction, is nearly impossible, right? So we can't see fusion, we can't measure shape because it's a Gaussian, it's the point spread function, and I'll come back to molecular mobility in a little bit. So okay, so one relatively easy experiment, though by no means perfect, and there are a lot of caveats, but one strategy that we took, because we sort of couldn't do things at this small length scale, was look at what is called hexane diol. So this is a solvent. It is known to disrupt weak hydrophobic interactions, and that just is because it was used to permeabilize the nuclear pore complex in previous applications. And so what I want to point out is that it is useful because it has been shown to dissolve some membraneless organelles, but is by no means a definitive experiment. It's not necessary or sufficient evidence for LLPS, but people have used it. And so, for example, in yeast, P bodies are a condensate, a liquid-like condensate, and if you mock treat cells, the condensates are stable. If you treat with hexane diol, they dissolve, and importantly, this is reversible. So if you wash out hexane diol, the P bodies can reform. But in contrast, if you look at a prion or an amyloid, so these are clusters of proteins that are not liquid-like, these are resistant to hexane diol, so they don't dissolve in the presence of hexane diol. And so people have started to use this chemical to distinguish between liquid droplets and solid aggregates. And so we tried to do this in our E. coli cells, looking at the RNA polymerase clusters. So in mock treated cells, you see the clusters are stable. When you treat with hexane diol, they disperse, and the polymerase distributes throughout the entire cell. And then when you wash them out, the polymerase can come back and cluster. So it is reversible, and it's certainly sensitive. And it's also interestingly, it's dose dependent. So if you add more hexane diol, then they dissolve more. And so that, to me, suggests that it is acting like a solvent, and not a stress response. I should also mention that the cells, this is a 5-minute treatment. So if you do it for much longer, the cells die. Like, they're not happy. Like, this is not a physiologically happy cell. But on short time scales, they dissolve. But critically, to compare with the sort of null hypothesis of DNA binding, I was really curious to see whether DNA binding proteins in general are also dissociate from DNA. And so to test that, we compared the RPOC to LAC-I. Again, these proteins are bound to a specific sequence in the DNA, and to the same protein, RPOC, so a large subunit of the polymerase, that is overexpressed. And these overexpressed proteins, rather than forming clusters in the nucleoid, it forms what are likely to be inclusion bodies at the cell poles and mid-cell. So the nucleoid here is actually excluding this overexpressed protein. And so what we found was that the DNA-bound LAC-I and the RPOC overexpressed inclusion bodies are resistant to hexane diol. So hexane diol itself is not sufficient to just dissociate proteins bound to DNA, and it also can't dissociate in bacterial cells either aggregates. And so there's something physically different about the clusters that RPOC forms relative to just DNA binding alone or to just proteins aggregating together. So again, this is consistent with phase separation, but is by no means definitive. And so if we come back to this question, we really wanted to do something that was definitive. And so we tried to look at molecular mobility. So before I go there, I'll pause to see if there are any new questions.
I think you're doing pretty well.
Okay.
Maybe
okay, yeah, I mean, they're definitely subdiffraction at the level of light microscopy, which was one of the reasons why we were trying to look into EM, but that is certainly not my forte, so it's a little slow. Okay, so to look at molecular mobility at this length scale in the sort of the subdiffraction regime, we turn to single molecule tracking. And so the idea here is that we can use fluorophores that you can either turn on, so photoactivate, or photoconvert. So we use a protein that fluoresces green, and then with UV, you can convert it to a protein that fluoresces red. And so this allows us to isolate individual molecules and visualize them in red, separate from the bulk in green. And so the basic premise is that we can, by isolating individual molecules and only visualizing one molecule at a time, we can very precisely determine the position of one molecule, and then we repeat this activation. So in one time step, we activate this molecule, and then we track that over time. And over here, we activate a different molecule, and so we can iteratively track single molecules over time, and then accumulate trajectories of single molecules over time in a cell. Okay, and so I should mention that this project was not only spearheaded by my graduate student Baljat Parmar, but also in collaboration with Rodrigo Reyes-Lamas at McGill, who basically showed us how to do single molecule tracking. So the idea of Baljat's experiment was we have cells, he activates them with UV light, and that turns on one or a few cells or few molecules in the cell, and then we track those over time. And then if we do this many times, then we start to see locations in the cell where molecules tend to stay, and other locations in the cell where molecules just freely diffuse. And so here is a movie of RPOC fused to M-maple, which is this photoconverting fluorophore, and you can see bright single spots that move over time, and so we're just going to accumulate many of these over time. And so Baljat can calculate the apparent diffusion coefficient for each trajectory, and what he sees is a very broad distribution. And it has a sort of long tail too. And so to try to understand to calibrate what this means, we compared this again to LAC-I. And so LAC-I is bound again to a single locus-specific sequence in the DNA, and it is at the sort of time scale that we're looking at it, we're not looking at unbound molecules, we're only looking at DNA-bound molecules, stably DNA-bound. And so the sort of lower limit of DNA mobility, so the DNA is jiggling around inside the cell itself, is around here. And so intriguingly, most of the polymerases move much faster than that. So they're not bound or stably bound to the DNA, but there is an overlap. And so what we were really interested in to see was, are these slow moving cells in everywhere, or are they distributed in specific locations in the cell? And so what Baljat found was by looking at the density of tracks and correlating that with the mobility or the apparent diffusion coefficient of each track, what you can see, I hope, is that in locations where the density of tracks is high, so these correspond to the clusters that we saw in the fixed cells, the molecular mobility is slow. And so the molecules that are in locations that have a high molecular density move slower than the average polymerase, and that this distribution, this red distribution, which is in the droplet or in the cluster, overlaps with the with LAC-I. So these proteins down here on the distribution are presumably directly bound to the DNA and involved in transcription. But what I was really excited to see was that there is another population of molecules that move more slowly than the rest of the population, but are still localized to these clusters, and they move faster than the DNA. And so we interpreted this as sort of molecules moving in a condensed phase. And so to sort of break open this distribution, so now what I'm doing is I'm taking the pink distribution here, polymerase, and I'm subdividing them by whether they are inside a local density with a minimum of a certain number of tracks throughout the movie. So that is represented by these in green molecules, and then blue molecules are always out. So they're always not in these high density regions. And then there are a few molecules that, that enter in and out of these defined clusters. And so what we can see is that in the in molecules, there is a bimodal distribution. And so these molecules, the slow fraction, are likely involved in transcription and actively transcribing and bound to the DNA. And then the molecules that are either moving in and out of these high density regions, high density clusters, are likely diffusing in a liquid droplet that is surrounding this DNA locus, but not freely diffusing. And then the molecules that are moving very quickly in the out fraction are either non-specifically bound to the nucleide or diffusing freely in the cytoplasm. And so there seem to be at least 3 distinct populations of RNA polymerase molecules. And so from that, it's hard for me, at least if you guys have any ideas, I'd love to hear, to sort of reconcile this intermediate mobility fraction with DNA binding. And so what we've concluded is that these represent polymerase molecules that are, condensed and moving inside a liquid droplet. And so, so what I've shown you is that clustering is rapid and reversible. The polymerase exhibits multiple mobility states, and even the slow states move more quickly than LAC-I. And that single molecule tracking, not only for bacteria, but even in eukaryotic systems, is a potentially powerful tool for detecting liquid-liquid phase separation, primarily at small length scales, but even at larger length scales, because FRAP can, differences in FRAP recovery rates can arise not only from phase separation, but also from changes in binding, on-off rates, et cetera. And so single molecule tracking may be able to resolve some of those open questions. So that is kind of where I'll leave it, and then I'm going to speculate for a little bit on what the function of this is. And so the elephant in the room of the phase separation field is really, what is the function of these condensates? And the hand waving explanation and the assumption that I think the field sort of assumes is that by concentrating molecules, you can increase the local concentration and thereby accelerate biochemical reactions, right? And so the hypothesis here is that by having the RNA polymerases sort of a pool of them locally around these genes, then maybe that can accelerate the reinitiation rate to maintain high levels of ribosomal transcription, ribosomal RNA transcription in fast-growing cells. And then if you didn't have them, these molecules would just diffuse away and throughout the cell. The truth is that these cells are so small, and the diffusion rates are, diffusion is a relatively efficient search strategy in bacteria, that I'm not sure that this is necessary. And so what we're doing now is trying to look at transcriptional output and initiation rates, et cetera, with and without clustering. So that's sort of one specific question: what is the function of these polymerase clusters in bacteria? But more generally, what are the function of condensates? And so I've been focusing on bacteria, but there is a direct analogy in eukaryotic cells. So Ibrahim Cisse has looked at Pol2, which transcribes mRNA genes in eukaryotes. And so Pol2 also forms clusters in the nucleus. There are clusters that are really transient, so they're small, they form, and then they disappear. And then there are larger, more stable clusters. And so the transient clusters are stable for about 12 to 13 seconds, and the large clusters are stable for minutes. And this is true not only for polymerase itself, but for several transcriptional co-activators. So mediator also forms condensate liquid-like clusters in the cell nucleus, and these are thought to co-localize with super enhancers and regulate the activation of particular sets of genes. And there again, because this is hard to do, but there is some evidence from Ibrahim's group that the clustering directly, corresponds to function and sort of stabilizing polymerase at the promoter and allowing initiation. And so the time scale of the cluster, or the duration of the cluster, longer clusters tend to correlate with more RNA. And so if you have a stable cluster, then maybe you can initiate successive rounds of transcription and control transcription by concentrating the polymerase at the promoter. And there's sort of active research going on in eukaryotes to try to really tease apart that functional consequence of the clustering. And then with that, I just want to point out that there are a number of other examples of phase separation that are coming out in bacteria. So not only the polymerase, RNase E is an RNase involved in mRNA degradation. It forms liquid-like clusters in Colobacter. PopZ forms condensates and sort of like a molecular mesh in the poles of Colobacter, which is important for the asymmetry of division. I mentioned storage granules at the beginning, but these poly-P granules, Lisa Racki has some beautiful EM snapshots that look a lot like Oswald ripening. And then there's a recent report on PAR-B showing that these may also form condensates to segregate plasmids and DNA. So with that, I will just thank Anne-Marie, postdoc who started this project, Baljot, who really took the single molecule stuff and was able to sort of look more beyond phenomenology, and then several undergraduates contributed to this work. And Rodrigo was amazing in helping us do the single molecule tracking. So I'll stop there, and hopefully there can be time for some discussion and questions.